So now that sequencing techniques have advanced, we can do a lot more with various different genomes. When we're talking about a genome, we're talking about a complete set of DNA in a single cell of an organism. When we're talking about genomics, then we're talking about the study of genomes, or usually through the sequencing of these entire genomes. So we can talk about things like structural genomics, functional genomics, comparative genomics, metagenomics, and others. We can use bioinformatics, which will use computer-based approaches to organize, share, and analyze the data. So much information is being known, or being isolated from these sequencing techniques and other things like that, but we really now need people who can start to analyze the data through using a lot of these different types of computer tools. So we can use these to look at things like gene structure, gene sequence, gene expression, what the resulting protein would look like, how it might function, and so on. GenBank, and you guys hopefully have used this now several times through the semester, is one of the most important genomic databases in the entire world. It is maintained by the National Center for Biotechnology Information, or NCBI. Every sequence that's deposited in GenBank receives an accession number. And so you can use these when you're, say, publishing a paper or something like that, so everybody knows what exact sequence that you were working with or that you received. Annotation is the process of identifying genes, their regulatory sequences, their functions, through that raw DNA sequence. And so now that the human genome has been sequenced, we're going to talk more about this later, there's still quite a lot of genomic information that has not been annotated correctly, or that has not been, the function of these genes has not yet been determined, or how they're regulated has not yet been determined. BLAST is another important tool, and this is just a software application that will allow you to compare different segments of DNA together. You can use genomic DNA from whatever organism you want. You can have, if you have found a clone, you found some cool DNA, you want to compare it just to see if there's anything else that's in a similar organism. Basically, it's just used to compare sequences throughout the major databases. And what you're looking for there are portions that are going to align with or that are the same as existing sequences. You can see what's the same and how many base pairs are different. What's changed? Are there any regions that have been added or been deleted, and so on. So this is an example of a query that you might run using BLAST. And the first is going to be the sequence. This is the query. The second is the subject. And what they did was they compared their particular sequence to mouse chromosome 8. And what they found was that the sequence is pretty similar. It's 93% identical, 0 gaps in there, so that means that there were no regions that were deleted or added in either of them. And most of these bases are matching up pretty well. The ones that don't are still going to be similar. You're going from a C to a G, here's a T to a C to a T. But from this, just simply this data, we can't say what sort of an impact that it might make on the resulting mRNA and the resulting protein from this. There are no deletions or additions, so there might not be any frame shift mutations. But there may be some single base pair changes. This can potentially change the codons. It may be a silent mutation. It's difficult to say without looking at the mRNA sequence from this. And one of the ways you can get this is through annotation. So when you're looking at these sequences, what you can do is you can try to identify open reading frames. And when you're looking at this, it really requires translation of all 6 potential reading frames. So if you have DNA, remember that these bases are going to be read in groups of 3, and there are 2 strands. So you can go one direction or you can go the other direction. So there's 2 different frames there. Each of those is a codon of 3, and if you shift it down 1 or shift it down 2, that could potentially give you a different reading frame. And so that's why there ends up with 6 potential reading frames from that. So here's a sample piece of DNA that someone maybe had sequenced. They're typically grouped in bases of 10. No real reason for that, except for that it's easier for your eye to kind of see and to follow and go through. Here's an example of that same sequence after it has been annotated. So a computer has gone through and identified, here's an ATG, here's a start site. This sequence seems to correspond to an exon. This sequence here seems to correspond to an intron. Here's the start of another exon. Here's another large intron. Here's another exon. May continue. Doesn't say for sure that there's a stop codon, although TAA is usually a stop codon, so my guess is it probably ends there. Up here, this is probably an upstream regulatory region that proteins might bind, or the regulatory proteins might bind to. So when we're looking at these open reading frames, or ORFs, they're just stretches of nucleotides that when you use the computer tool, you can translate it to protein through this computer analysis. And you can figure out what would this sequence be, where would this stop be, and so on. So here's a potential sequence here. It's Homo sapiens, tuba 3C, and it gives you just the base pairs 1 through 300. So these are all of the predicted polypeptides from that particular sequence. Looking at one of these strands, 5' to 3', if you're looking at frame 1, there's a stop codon right away. There's another stop codon right away. Okay, there's a lot of stop codons here, not a lot of M's, so probably this is not going to be a polypeptide from this region. Maybe it's a short polypeptide, but probably not. If you look in frame 2, similar thing. Okay, we get some stops. There are some methionines after this, so it's possible that there's a protein that starts down here, or a protein that starts here, and we don't have any further sequence, so it's really difficult to say whether or not that would continue or whether that would stop. In frame 3, there's a methionine, there's another potential start methionine, another potential start methionine, no stop sequence, so this could potentially be a protein product, and it's suggestive of a protein coding gene. The opposite strand, there's immediately a stop, methionine, a stop right away. There's a methionine down here though, with no stop codon after it, and it's possible that this here could potentially be a polypeptide in that direction. The 3' to 5', there's a lot of stops here, probably not a protein coding gene. The 3' to 5', frame 3, this potentially also could be a protein coding product. There's a start methionine and no stop codon within this region. If you kept looking at the sequence, it's possible that this is a protein coding gene. So the reason why I went through all this seems kind of boring, but this is basically what the computer tools will do. They'll go through a particular region, they can predict that there's a polypeptide there, but it really takes a human kind of thinking about it. Is this suggestive of a protein coding gene or not? What more information do we need to make that determination? Do we need to clone this into bacteria to see what kind of product it makes? That might be a step that someone takes if they see something that looks like a putative gene. In functional genomics, what we're doing is looking at the study of gene functions basing on the RNAs or possible proteins that they code, as well as their regulatory elements. So you're using your gene sequence and you're predicting that polypeptide sequence. You can then analyze that polypeptide or amino acid sequence for specific known protein domains and motifs. So there are known protein domains such as ion channels, membrane spanning regions, secretion signals, export signals, and so on. If your protein, your putative protein, contains some of these, it might give you a hint at the function of that particular protein. There are also known motifs, things like a helix turn helix, a leucine zipper, zinc finger motifs. These are all known to bind either DNA or bind other molecules. And again, it can give you a hint of what the role of that particular protein might be. So this is Arabidopsis. This is a plant that's widely used in genetics. The genome of Arabidopsis has about 25,000 open reading frames. It's about 125 megabases, and this is how those genes are classified. So there's a number of them that are known to be classified with metabolism, transcription, cell growth, cell division, and so on. But there's a huge number of these genes still, about a third of the genes or so, or a little more, that are unclassified. And so they're putative genes, but we just simply don't know what their functions are at all. Functional genomics. BLAST sequences can be used to screen databases to compare a sequence to a known sequence. And so through this, say you have a sequence, you don't know what it's for, but what you can do is you can screen it against organisms, see if something similar pops up. We might know what the function is of that gene in a different organism. It might have the similar function in that other organism. So you can use these similarity searches to find homologous genes. They're going to be evolutionary related in other organisms. So for example, you can see here, this is a human leptin gene, and this is a gene that's involved with controlling hunger, as well as feeling full. It's homologous in mice here, and what you can see is it's pretty darn similar. There's a few spaces, here's an extra codon here, there's a few base pair changes here and there, but throughout it's very, very similar. So we can study this gene in mice, and theoretically at least, the human genome is going to have a similar function to what we learn, and what we learn in these mice should be applicable to humans as well. If you want to study how proteins and DNA interact, there are several different techniques that you can use. One of the reasons why you might want to see something like this is to see whether or not a particular protein is a transcription factor and whether or not it binds DNA. So you can use chromatin immunoprecipitation or CHIP assays. What you do first is you isolate DNA and you cross-link, you add formaldehyde to cross-link these proteins to the chromatin. So normally, if you're doing these different manipulations, these proteins would not be stuck to the DNA like this, but this formaldehyde is going to make them stick to this DNA. You extract the DNA, you shear it, you break it into pieces, you take out antibodies, which are going to be coupled to beads, which are going to be specific for the protein that you want to study. In this case, it looks like it's looking for this pink protein here. And you can specifically pull out that pink protein along with any DNA that happens to be bound to it. You can purify the protein, or excuse me, purify the DNA from here. You can amplify it through PCR, and at this point you can either run these samples on a gel, and this is where you would pretty much end up if you were doing a chip. You can also do a chip-on-chip, and you can look to see all of the different places where this happens to be binding. All of the different DNA sequences for which your protein of interest happens to bind. So a pretty cool technique to look at what genes a particular transcription factor will control.